Okay, so welcome to this next video in which we are discussing the Wnt beta-catenin pathway, also known as the canonical Wnt pathway. Okay, right, so we're currently looking at what is happening within the nucleus when the Wnt beta-catenin pathway is in the off state. So remember, in the off state we have very low levels of beta-catenin, okay, and in the absence of beta-catenin, what happens is you have these TCF-LEF transcription factors binding to these Wnt response elements, which are these sequences of 8 organic bases which you have in a huge number of different promoter regions. Okay, and these proteins remain bound to those Wnt response elements, or those WREs, all the time. Now, when the beta-catenin is present, what will happen is beta-catenin will bind on top of the TCF-LEF family transcription factor. But at the moment we're in the off state, so there is no beta-catenin present. So instead what's going to happen is a Groucho-TLE family protein is going to bind on top of the TCF-LEF family transcription factor. And this Groucho-TLE family protein is then going to bind to a histone deacetylase, okay, which now is going to remove acetyl groups from the lysine residues within the histone proteins. Okay, so let's just discuss the acetylation of lysine residues within the histone proteins. So, basically, if you want to express a gene, okay? If you want to actually make mRNA for a gene, and your gene is wrapped around a histone core complex, which is very likely because this is how DNA exists, the entire chromosome will be like this, basically. Most of the DNA is actually going to be wrapped around histone core complexes, which means that it's most likely that at least part of the gene is going to be wrapped around a histone core complex. If you actually want to make mRNA that corresponds to that gene that's wrapped around the histone core complex, you need to loosen how tightly the DNA is wrapped around the histone core complex, because when it's wrapped very tightly, the machinery that's going to make the mRNA can't get access to the DNA easily. Okay, so one way that you can loosen up the attachment of the DNA to the histone core complex is by acetylating lysine residues within the histone proteins. Okay, and that makes the DNA loosen how tightly it is coiled around the histone core complex, and that opens the DNA up so that now transcriptional machinery can work its way along that DNA and make mRNA. Okay, so let's discuss the acetylation of lysine residues within proteins. So let me start by drawing a lysine residue. So here's the amino group, drawn as though it's bound to the carboxylic acid group of the amino acid prior to it. Here's the alpha carbon with the hydrogen coming off. Here's the carboxylic acid group, which will be bound to the amino group of the next amino acid along. Okay, now the R group of lysine consists of 4 methylene groups. Now I don't have that much space, and I want to show an acetyl group bound to the amino group. So what I'm going to do is I'm going to show the 4 methylene groups just as one methylene group, like so, bracketed, and then I'll suffix it 4, which means repeat this methylene group 4 times, basically. Okay, that's a useful trick. Now, then we have an amino group right on the end of the 4th methylene group. Okay, and that then is the structure of the amino acid lysine. Now, lysine residues within certain of the histone proteins in the histone core complex are going to be acetylated. So what does it mean to acetylate a lysine residue? Well, it means that we're going to add an acetyl group onto that lysine residue. So what is an acetyl group? Well, to understand that, we need to know what acetic acid is. Okay, so I think I'll squeeze acetic acid up here. Okay, so acetic acid is the old name for a molecule that would now be called ethanoic acid. Okay, and it's just a 2-carbon carboxylic acid that is fully saturated. Okay, like so. Very simple. Okay, so it's the acid that's within vinegar, for instance. Okay, right. So what we're going to do is we're going to react this acetic acid molecule with the amino group of the lysine residue to form an amide link. Okay, so let me put my acetic acid molecule here. Okay, so I'm just flipping it round. So here's the carboxylic acid group, and here is the methyl group coming off the carboxylic acid group. And what we're now going to do is create an amide link between the carboxylic acid group of the acetic acid molecule and the amino group of the lysine residue. So what happens in the formation of an amide link is you take off the alcohol group from the carboxylic acid group, you take off the hydrogen from the amino group, okay, you combine those two things together to make water, okay, and you then bind the nitrogen of the amino group to the carbon of the carboxylic acid group. And this link that we've now got here, this new link that links the acetic acid molecule to the amino group of the lysine, this is what's known as an amide link. Okay, right. So why is this process known as acetylation? Well, basically, what is an acetyl group? Well, hopefully you can see that we have not actually added on the full carboxylic acid molecule. We've not added on the entire acetic acid molecule. We've got very close. We added on all of the acetic acid molecule bar the alcohol group that comes off the carboxylic acid group. Okay, now the name for this structure that I've circled in green here, which is not a molecule because this carbon needs another bond, okay, so it has to be part of a larger molecule, this is known as an acetyl group. So remember, a group is part of a molecule, okay, so this is not a molecule in itself because this carbon needs another bond, so it's a group instead, an acetyl group. Okay, so that's why this is known as acetylation of the lysine residue, because we're effectively adding an acetyl group onto the lysine residue. Okay, so basically, certain enzymes can add acetyl groups onto lysine residues in certain of the histone proteins, okay? And this causes the DNA to loosen how tightly it is wrapped around the histone core complex, okay? And that means that that DNA can be transcribed more often so we can use it. So, acetylation of lysine residues within the histones is a marker that that portion of DNA is actually active. It's actually being used for transcription. Now, we have just attached onto our TLE Gaucho family protein a histone deacetylase. So this is an enzyme which actually removes acetyl groups from histones. Okay, so it is now going to take acetyl groups off the histones. So what's then going to happen is the DNA in this local region now, close to where this histone deacetylase is bound, is now going to coil more tightly around the histone core complex and is going to stop being expressed. So this is actually going to be repressing the transcription of the downstream gene, basically. So if you've got a promoter region which has one of these Wnt response elements within it, then what's going to happen is you're going to assemble this complex. The histone deacetylase is now going to deacetylate the local histones, which is then going to cause the DNA to wrap more tightly around the histone core complexes, and that means that the downstream gene is actually going to be less likely to be transcribed. So at the moment, this transcription factor is acting as a transcriptional repressor. Okay, so remember, transcription factors can be enhancers and also repressors. They can either increase the expression of the downstream gene by increasing its transcription, or they can decrease the expression of the downstream gene by decreasing its expression. And we've now seen a mechanism by which this transcription factor here is now going to decrease the expression of the downstream gene. Okay, now let's return over to talking about the on state. Okay, so we know in the on state of the Wnt beta-catenin pathway, what's going to happen is Wnt arrives in the extracellular fluid, it activates the LRP5/6 receptors along with the Frizzled receptors. That causes the inactivation of the beta-catenin destruction complex, and then beta-catenin survives. It goes up within the cytoplasm, okay, and then beta-catenin molecules start moving into the nucleus. So it's finally time to discuss the concept of a transcriptional coactivator. So beta-catenin itself is not a transcription factor. It's one step further along than a transcription factor. Okay, so the transcription factor is still going to be this TCF-LEF family protein, okay? And remember, we've seen 4 members of that family. Well, the 4 members of that family. So here is our TCF/LEF transcription factor, which, remember, is bound to that sequence of 8 organic bases, which is CC, okay, then TTT, okay, then GWW. Okay, and I'm only showing one side of it, but of course there will be the complementary strand as well. So that's the Wnt response element, the WRE here. Okay, so let's colour in the WRE, which is contained within our promoter region. Okay, so in fact I might say that this larger portion here, this is the promoter region of some downstream gene over here. So let's say the gene is here. Okay, so I'll just extend the DNA a little bit. Okay, so this is our gene down here, and we'll highlight the gene in red. And then upstream of the gene, we have this promoter region, which contains a Wnt response element here. So all of this is the promoter region. Okay, which I'll highlight in turquoise here. Okay, right. And finally, we'll also colour in our TCF-LEF transcription factor, which will be in blue here. So that's the transcription factor that's binding to the promoter region. Now, a transcriptional coactivator then is a protein which can bind on top of a transcription factor. Okay, and beta-catenin is going to bind on top of this transcription factor here. So it's going to be a transcriptional coactivator. And then this protein which binds on top of this transcription factor is now going to completely change the function of this transcription factor. Before beta-catenin was here, TCF/LEF transcription factors were resulting in the downstream gene being less likely to be transcribed. Okay, so they were transcriptional repressors. Now, once the transcriptional coactivator beta-catenin has bound, you're actually going to turn this transcription factor from being a repressor to being an enhancer, basically. Okay, so the transcriptional coactivator is going to bind to the transcription factor and change the function of the transcription factor, changing it from being a repressor to being an enhancer. So now this is going to increase the expression of the downstream gene, and I want to discuss how. Okay, so on top then of beta-catenin, loads of other things are now going to bind, okay, but an important one that we're going to discuss is that you're now going to get enzymes known as histone methyltransferases binding. Okay, so instead of histone deacetylases now, we've got histone methyltransferases. Okay, and for short, histone methyltransferases are often abbreviated to HMTs, H for histone, M for methyl, T for transferase. Okay, so we'll have our histone methyltransferase here in red. Okay, now, this enzyme is once again going to add groups onto lysine residues within histone proteins. Okay, but this time we saw previously that the histone deacetylases were removing acetyl groups from lysine residues of histone proteins. Histone methyltransferases are going to add methyl groups onto the lysine residues of histones. Okay, so what we're now going to do is, if I draw the lysine residue again, okay, I'm now going to draw the lysine residue with a methyl group added onto it. Okay, so here's the core amino acid structure. Then the R group consists of, once again, 4 methylene groups. Okay, so here's one methylene group, and I'll then bracket it and put a 4 there. Okay, and this time what we're going to have is a methyl group added on to this nitrogen atom here. Okay, now usually when you start methylating the amino group of lysine residues in histones, you end up adding multiple bonds onto the nitrogen. Okay, so previously, remember, the nitrogen only had 3 bonds, and that's how nitrogen likes to be. It only likes to have 3 bonds. Okay, but when you add these methyl groups on, you're going to move the nitrogen up to having 4 bonds. So that's not what nitrogen likes to have. Nitrogen likes to have 3 bonds. So when you make this 4th bond here between the nitrogen and the methyl group, what's actually going to happen is the nitrogen is going to put both electrons into this covalent bond. So both of the electrons in this covalent bond will come from the nitrogen. So it will put its lone pair of electrons into this bond. Now, the understanding of a covalent bond is that one electron belongs to each of the members within that covalent bond. So when the nitrogen does this, it's effectively like the nitrogen has donated an electron to the carbon, because the understanding now is one of these belongs to the nitrogen and the other belongs to the carbon. So the nitrogen has effectively donated an electron away, which gives it a positive charge. Okay, you might then ask, so does the carbon have a negative charge? Okay, and the answer is no, and the reason is that when it came into this sorted arrangement here, the carbon will have originally had a positive charge, so when it's received this electron from the nitrogen, it's become neutralized, okay, and it's effectively deferred the positive charge onto the nitrogen. Okay, so that's how you methylate lysine residues within histones. Now, what's the significance of the methylation of lysine residues within histones? Well, once again, if you methylate lysine residues within histones, the DNA, just like acetylation, is going to wrap around the histone core complex less tightly. Okay, so it's going to loosen up how tightly it wraps around the histone core complex. Then that makes the DNA more accessible for transcription, so transcription is going to go up. So basically, the assembly of this complex on top of this Wnt response element within the promoter region is going to result in the downstream gene being transcribed more often, because the DNA here is going to be wrapped around histone core complexes less tightly, basically. Okay, so now what we're going to do is increase the expression of all of the genes which have promoter regions upstream of them which contain a Wnt response element, like so. Okay, so that is basically what the activation of the Wnt beta-catenin pathway does. It's going to increase the expression of all of the genes which have a Wnt response elements within their promoter regions. Okay, so overall it's going to change gene expression. It's going to upregulate the expression of a huge number of different genes. Now, generally, the genes that you turn on are the genes that you need in order to divide, basically, in order to grow and divide. So I told you right at the beginning that the Wnt beta-catenin pathway is involved in controlling a cell and making it divide, basically. Okay, so let's just have a brief discussion of the cell cycle then, and what the activation of the Wnt beta-catenin pathway within a cell is going to do. Okay, right. So, we're going to start off with a cell that is sitting quiescently. Okay, so this cell is perfectly happy, it's not dividing, and when a cell is in the quiescent period, it's set to be in the G0 phase of the cell cycle. Okay, so this is not an active portion of the cell cycle. This is a portion where the cell is not dividing, basically. So most of the cells in your body are not dividing. Those are in G0 phase of the cell cycle. Okay, then when a cell receives signaling via, for instance, the Wnt beta-catenin pathway, what's going to happen is it is going to now move from G0 phase to an active phase of the cell cycle, known as the G1 phase. Okay? So it is this transition here between the quiescent phase, where you are not getting ready to divide and you are just sitting doing whatever a cell does, okay, and the active phase, where you're actually going to start getting ready to divide. It's this transition here that the Wnt beta-catenin pathway is going to be involved. Okay, so this change in gene expression that you get as a result of activating the Wnt beta-catenin pathway, this is going to result in the cell moving from G0 phase to G1 phase. Now, what occurs in G1 phase? Well, basically, the cell starts growing, it gets ready to divide. So, basically, in order to split in two, the cell firstly needs to get big enough to split into two cells, because if it just divided now, each of the cells would be half the size of the original cell. So, firstly, the cell has to actually enlarge itself so that it's big enough to divide into two cells. Okay? So, basically, what needs to happen is you are going to be starting to duplicate intracellular organelles, and the exception is the nucleus. The nucleus, we're going to study the nucleus division separately. It's going to occur much later on, but the other intracellular organelles, they're all going to be duplicated. In addition, all of the essential proteins of a cell are going to need to be duplicated as well. Okay, so we're going to need to be making lots more protein. All of the enzymes involved in respiration, all of the receptors, all of the signaling pathways, all of these proteins, these essential proteins, you know, the cytoskeletal proteins as well, all of them need to be duplicated. You need to make more of them, okay? And to make more protein, you need to activate the transcription of the genes for these proteins, and that's what we're doing here. We are activating the transcription of the genes that are going to be involved in making the cell bigger, basically. Okay, in addition, in G1 phase, you prepare to copy the DNA, okay? So, in the nucleus of a cell, you have 46 chromosomes, okay? And every single one of those chromosomes is going to need to be copied so that the two daughter cells can each have a copy of each of the 46 chromosomes. So I'm just going to draw one chromosome, but this will represent the 46 basically. So here, let's say this is chromosome 1. Okay, one of the chromosome ones, because of course you have two chromosome ones that are homologous. They're not identical, but they are homologous. They have the same sort of genes on them. Okay, one is maternal, one is paternal. Okay, right. So this is a chromosome that's going to represent what's going to happen to all 46, because I don't want to have to draw 46 of them out. Okay, right. So you're going to get ready to replicate the DNA. You're going to start making the machinery and preparing the machinery that's going to actually be involved in copying the DNA. Then you're going to go into the next phase of the cell cycle, which is known as the S phase of the cell cycle. Oh, and by the way, I didn't tell you what G1 actually stands for. This stands for the first gap or first growth phase. Okay, so it's known as a gap or a growth phase. Okay, this is the first one. There is going to be a second one. Okay, the S phase stands for the synthesis phase, and in the synthesis phase of the cell cycle, what's going to occur is you are going to copy all of the DNA. Okay, so we've now got our large cell here. Okay, here is the nucleus, and we're going to be copying every single chromosome. So here is one chromosome, we're going to copy another one. Okay, so each of the 46 chromosomes in this nucleus is going to get replicated. Here is the identical copy of the one of the 46 chromosomes that I have shown here. Okay, and these two copies that we now have are known as sister chromatids. Okay, so they are absolutely identical, basically. Okay, right. So, then what's going to happen is we're going to go into a phase known as the G2 phase, or the second gap or growth phase. Okay, and in this phase, the cell is going to get even bigger. Okay, so let's show this. So it's going to grow even more. Okay, it's got its nucleus that has twice as much DNA as it needs, basically. It's got 2 copies of all 46 chromosomes, okay, which are paired together as sister chromatids, and it's going to grow even more. It's also going to prepare for the final phase of the cell cycle. And by the way, I think I'll add a little bit of colour onto this. So here is the G1 phase in green here. Okay, here is the S phase in blue here. Okay, here is the G2 phase in purple here. Okay, and then the final phase is going to be the M phase, okay, which will take you back to here, basically. And if the signal to divide is still present, if the Wnt is still present in the environment, what will then happen is you'll go through the cycle again, basically. It will take you into G1 phase once again, and you'll go round and round, why it's known as the cell cycle. Okay, right. So this final phase of the cell cycle is what's known as M phase. However, M phase can really be divided up into 2 separate important portions. Okay, the first portion is mitosis. Okay, so let's colour in mitosis in orange. Now, mitosis, strictly speaking, means nuclear division. Okay, it does not mean cell division. That's a common misinterpretation. Okay, so what's now going to happen in the mitosis portion of M phase is you are going to split this nucleus, which has twice as many chromosomes as it needs to have, into 2 identical nuclei, which then have 1 copy of each of the 46 chromosomes. Okay, so here is our gigantic cell now. Okay, and what you're going to do is split the nucleus into 2 nuclei. Okay, and each one of these will have 1 copy of all 46 chromosomes. So you're going to separate the 2 sister chromatids for all 46 chromosomes. One of them will go into 1 nucleus, so let's say this is one of them in purple, and the other one, here in green, will go into the other nucleus. So by the end of mitosis, you've got this gigantine cell which has got 2 identical nuclei within it. Okay, and then the final portion of M phase, which is in red here, okay, is known as cytokinesis. Okay, and this then is the division of the cell that has now 2 nuclei within it into 2 separate cells. Okay, so by the end of cytokinesis, what we're going to have is these 2 separate cells which have both got nuclei within them which are identical. Okay, so here is the nucleus. Okay, and each of them has got 1 copy of all 46 chromosomes here. So cytokinesis is the strict name for cellular or cytoplasmic division, like so. Okay, and as I say, if the Wnt signal is still there, these cells will go straight back into G1 phase. They'll start growing, and then they'll copy their DNA and they can go round and round. If the Wnt signal is gone, what will instead happen is they'll go down G0 phase here. Okay, they'll go into this portion here, and they'll go quiescent. Okay, right, so that then concludes our discussion of the Wnt beta-catenin pathway.